Chapter 1. Elements of the immune system and their roles in defense. Let's dive into the building blocks of immunity and discover how your body fights off threats every day. One of the greatest triumphs in medical history is the global eradication of smallpox. This chart shows the dramatic decline in the number of countries reporting one or more cases per month from 1965 to 1980. Through coordinated vaccination efforts and surveillance, the disease was steadily eliminated. By the late 1970s, the number of affected countries dropped to zero, and in 1980, smallpox was officially declared eradicated by the World Health Organization. This moment marked a defining success for the immune system's partner, modern immunization. This haunting image reminds us of the human cost of infectious disease. Here, a young child infected with smallpox clings to his mother, a stark illustration of the suffering caused by the virus before eradication. The image serves as both a warning and a tribute, a warning of what happens when disease goes unchecked, and a tribute to the global collaboration and scientific advancement that made eradication possible. The immune system, when supported by vaccination, can achieve extraordinary victories. Our bodies are home to trillions of microorganisms, but not all of them are harmful. In fact, numerous commensal microorganisms live peacefully within healthy humans. They colonize our skin, gut, mouth, and other body surfaces, forming a first line of defense by outcompeting potential pathogens. This symbiotic relationship plays a crucial role in maintaining immune balance and helps train the immune system to distinguish friend from foe. In a healthy colon, large numbers of commensal bacteria populate the gut lumen. They act as guardians, competing with harmful microbes for space and nutrients. When antibiotics are introduced, many of these beneficial bacteria are wiped out. This creates an opportunity for pathogenic bacteria to invade. Once they gain a foothold, these pathogens release toxins that damage the gut's epithelial lining, leading to mucosal injury. As the damage progresses, red and white blood cells begin to leak into the gut between the injured epithelial cells, a sign of inflammation and immune activation. This cascade highlights the critical role our microbiota play in maintaining gut health, and how the immune system responds when that balance is disrupted. Pathogens are infectious organisms that cause disease. Pathogens come in many shapes and sizes, and this slide shows a variety of infectious organisms under the microscope. Images A and B depict viruses, such as influenza and herpes viruses, microscopic agents that hijack our cells to replicate. See-through eyes show different types of bacteria, including Staphylococcus, Salmonella, and Mycobacterium tuberculosis, some of the most notorious causes of human disease. In J, we see a fungus like Candida, which can cause infections, especially in immunocompromised individuals. K shows protozoa, such as Trypanosoma, which causes African sleeping sickness. And finally, L reveals parasitic worms, large, multicellular invaders like those responsible for diseases such as schistosomiasis. Each pathogen presents a unique challenge to the immune system, and understanding them is key to understanding immunity. This table highlights a variety of viral pathogens and the diseases they cause, along with their classification and typical route of infection. For example, SARS is caused by a coronavirus and spreads through oral, respiratory, or ocular mucosa. Diseases like West Nile encephalitis and yellow fever are caused by flaviviruses and are transmitted through the bite of an infected mosquito. Sexually transmitted viruses like hepatitis B and HIV are part of the hepatinovirus and retrovirus families, respectively. Many common illnesses, like chickenpox, influenza, measles, and mumps, spread via the oral or respiratory route. demonstrating how quickly these pathogens can move through populations. Even diseases like rabies and AIDS, though transmitted differently, are viral in nature, showing the wide range of viral strategies for infection and survival. This diversity emphasizes the need for an adaptable and dynamic immune system. This table outlines a range of bacterial diseases, along with their causative agents, classification, and modes of transmission. Gram-negative bacilli are responsible for conditions such as bacillary dysentery, typhoid fever, and plague, often transmitted through contaminated food, water, or vectors like fleas. Diseases like gonorrhea and meningococcal meningitis are caused by gram-negative cocci, while Haemophilus influenzae and Legionella pneumophila, both Coccobacilli, can lead to serious respiratory infections. Gram-positive bacilli, such as Bacillus anthracis and Clostridium tetani, enter through spores or wounds, causing anthrax and tetanus, respectively. Gram-positive cocci, like Staphylococcus aureus and Streptococcus species, can cause infections ranging from pneumonia to tonsillitis. Other notable bacteria include Mycobacterium tuberculosis, Borrelia burgdorferi, which causes Lyme disease, and Treponema pallidum, the causative agent of syphilis. Each pathogen has evolved specific mechanisms to enter the body and evade immune defenses, challenging the immune system to adapt accordingly. This chart highlights pathogens beyond viruses and bacteria, including fungi, protozoan parasites, and helminth worms. Fungal infections such as aspergillosis, athlete's foot, candidiasis, and fungal pneumonia are caused by Ascomycetes fungi. These are often opportunistic pathogens, taking advantage of weakened immune defenses or entering through direct contact or inhalation of spores. Protozoan parasites include Leishmania, Plasmodium, Toxoplasma, and Trypanosoma species. They're responsible for diseases like leishmaniasis, malaria, toxoplasmosis, and African sleeping sickness, typically transmitted by insect vectors or contaminated materials. Finally, helminth parasites, commonly known as worms, include Ascaris lumbricoides, the common roundworm, and Schistosoma monsoni, a blood flu. These are transmitted either orally through infected materials or directly through the skin. Together, these diverse organisms represent the wide range of challenges the immune system must defend against. The skin and mucosal surfaces form barriers against infection. The body's first line of defense against infection is its physical and mucosal barriers. The skin forms a strong, protective outer layer, supported by hair and nails, which prevents most pathogens from entering. Inside the body, mucosal surfaces line areas like the respiratory, gastrointestinal, and urogenital tracts. These tissues aren't just passive barriers, they actively trap and neutralize microbes through mucus, enzymes, and immune signals. Even the eyes and mammary glands are protected by specialized secretions that help fight off infection. Together, these surfaces create a coordinated, intelligent shield that guards the body from the outside world. The innate immune response causes inflammation at sites of infection. When bacteria enter the body, the innate immune system is the first to respond. It begins when bacterial surfaces trigger the activation of complement proteins in the blood. One fragment of complement binds directly to the bacterium, while another acts like a beacon, attracting nearby immune cells. An effector cell, such as a macrophage, recognizes the tagged bacterium through its complement receptor. Once contact is made, the effector cell engulfs the invader, enclosing it within a compartment called a phagosome. Inside the cell, the bacterium is killed and broken down. A rapid, non-specific defense that marks the hallmark of the innate immune response. This early response not only clears infection, but also sets the stage for inflammation, and the activation of more complex immune pathways. In healthy skin, the immune system stays on alert, but the tissue remains calm and uninflamed. When a surface wound introduces bacteria, resident effector cells in the skin spring into action, recognizing the invaders and releasing signaling molecules called cytokines. These cytokines trigger vasodilation and increase the permeability of nearby blood vessels, allowing immune cells, proteins, and fluid to flood the infected area. As the immune response intensifies, the tissue becomes visibly inflamed. This results in redness, heat, swelling, and pain, the classic signs of inflammation, as the body fights to contain and eliminate the infection. The adaptive immune response adds to an ongoing innate immune response. The immune system operates through two interconnected branches: innate and adaptive immunity. Innate immunity responds within hours. Its recognition mechanisms are fixed and rely on a limited number of pathogen-detecting patterns. This response is fast and consistent, but not highly specific. In contrast, adaptive immunity takes longer to activate, typically days to weeks. It's slower but much more specialized, with an enormous range of highly selective receptors that improve as the response continues. Despite their differences, both systems share common effector mechanisms, working together to destroy pathogens and protect the body. The innate system buys time, while the adaptive system brings precision. The adaptive immune response begins long before infection ever occurs. During development, the body produces a vast library of lymphocytes, each with a unique receptor designed to recognize a specific target. Most of these cells remain inactive, waiting for a match. When infection strikes, only the lymphocytes whose receptors match the pathogen are activated. This process is highly selective, like finding the perfect key for a lock. These activated lymphocytes rapidly multiply and mature into effector cells. Their mission is clear: seek out and destroy the invading pathogen. Through this targeted activation and expansion, the adaptive immune system mounts a powerful, precise response to eliminate threats that the innate system alone cannot contain. Adaptive immunity is better understood than innate immunity. This graph illustrates the critical importance of both innate and adaptive immunity during infection. In a person lacking innate immunity, represented by the red curve, microbial numbers rise uncontrollably. Without this early line of defense, the infection becomes rapidly overwhelming, often with fatal consequences. In contrast, individuals missing adaptive immunity, shown by the green curve, can initially contain the infection thanks to their innate defenses. But over time, they struggle to fully eliminate the pathogen, leading to chronic infection. In normal individuals, shown in yellow, innate and adaptive systems work together. The innate immune response slows the infection early on, while the adaptive response builds up, targets the invader precisely, and clears the threat. This figure shows why both arms of the immune system are essential and how they cooperate to protect the body effectively. Immune system cells, with different functions, all derive from hematopoietic stem cells. All immune system cells originate from a common source: hematopoietic stem cells in the bone marrow. From this shared origin, a diverse array of highly specialized cells emerges, each playing a unique role in immune defense and tissue maintenance. Small lymphocytes include B cells, which produce antibodies, and T cells, which carry out helper and cytotoxic functions. Plasma cells are fully differentiated B cells that secrete large amounts of antibodies. Natural killer cells target and destroy virus-infected cells without needing prior activation. Dendritic cells initiate adaptive responses by presenting antigens to T cells. Mast cells release histamine and other mediators to expel parasites and trigger allergic responses. Monocytes circulate in the blood and mature into macrophages, which engulf microbes and coordinate immune signaling. Neutrophils are fast-acting phagocytes that are often the first to respond to infection. Eosinophils and basophils release granules to fight parasites and regulate allergic reactions. Outside of immune defense, megakaryocytes generate platelets for blood clotting and wound repair, while erythrocytes, or red blood cells, carry oxygen throughout the body. Together, these cells form the workforce of the immune system, each with a distinct task, but united in the defense of the body. The immune system's strength lies in its diversity of cell types, each originating from hematopoietic stem cells in the bone marrow. Small lymphocytes develop into either B cells, which produce antibodies, or T cells, which perform cytotoxic or helper roles. Plasma cells are the fully differentiated form of B cells, specialized in secreting large quantities of antibodies to neutralize pathogens. Dendritic cells act as sentinels, capturing antigens and presenting them to T cells, initiating the adaptive immune response. Mast cells, found in tissues, release histamine and other granules, helping expel parasites and contributing to allergic reactions. Each cell is finely tuned to perform a specific role, and together, they orchestrate a coordinated and powerful immune defense. From a common hematopoietic stem cell origin, more immune defenders emerge, each with a targeted function. Natural killer cells act quickly, identifying and destroying cells infected by certain viruses. They require no prior activation, making them a critical part of early antiviral defense. Monocytes circulate in the bloodstream as precursor cells. Once they enter tissues, they mature into macrophages, versatile cells that engulf and destroy microorganisms and initiate immune responses by activating T cells. Neutrophils are short-lived but highly effective. They swarm to infection sites and perform phagocytosis, rapidly clearing invading microbes. Together, these cells form the bridge between the rapid innate response and the more specialized adaptive response, working constantly to contain and eliminate threats. The final group of immune-derived cells demonstrates just how versatile hematopoietic stem cells truly are. Eosinophils specialize in attacking parasites that have been tagged by antibodies. They release toxic granules to kill these large invaders. Basophils, though few in number, help regulate immune responses to parasites, especially during allergic reactions. Megakaryocytes are responsible for producing platelets, tiny cell fragments essential for blood clotting and wound repair. And while erythrocytes, or red blood cells, aren't immune cells, they share the same stem cell origin. Their job is to transport oxygen throughout the body, supporting the energy demands of every cell, including those in the immune system. From defense to repair to circulation, these cells reflect the remarkable range of functions born from a single stem cell lineage. The production of immune cells, known as hematopoiesis, shifts across different organs throughout development. In early embryonic life, hematopoietic activity begins in the yolk sac, where the first blood and immune cells are produced. By around 3 months of gestation, this activity transitions to the fetal liver and spleen, which take over as the primary sites of immune cell development during mid-gestation. As birth approaches, the role of bone marrow becomes dominant. It gradually replaces the liver and spleen and continues to serve as the central hub for blood and immune cell formation throughout life. This shift ensures that as the body grows, its immune system has a stable, lifelong source of new cells ready to protect and repair. All immune system cells trace back to a single origin, the hematopoietic stem cell, a multipotent cell found in the bone marrow. From this one source, two major lineages emerge, the common lymphoid precursor, and the common myeloid precursor. Lymphoid precursors give rise to B cells, T cells, and natural killer cells. B cells can further differentiate into plasma cells, while T cells become effector T cells that coordinate or kill. Myeloid precursors generate a wide range of cells, including monocytes, which can mature into macrophages and dendritic cells, key players in both innate and adaptive immunity. They also give rise to granulocytes such as neutrophils, eosinophils, and basophils, each specialized for different forms of microbial attack or allergy regulation. A separate myeloid branch leads to megakaryocytes, which form platelets, and erythroblasts, which develop into red blood cells or erythrocytes. This beautifully branched lineage illustrates how a single stem cell gives rise to the entire cellular force that powers immune defense, blood transport, and tissue repair. Leukocytes, or white blood cells, come in many forms, but they aren't equally represented in the bloodstream. Neutrophils are by far the most abundant, making up between 40 to 75% of all leukocytes. They're the frontline defenders, rapidly responding to infection and inflammation. Lymphocytes, which include B cells, T cells, and natural killer cells, account for 20 to 50%. They provide the precision and memory of the adaptive immune system. Monocytes, which can differentiate into macrophages and dendritic cells, make up 2 to 10% of the circulating pool. Eosinophils, specialized in fighting parasites, appear at levels between 1 and 6%. And basophils, involved in allergic responses and parasite control, are the rarest, less than 1%. These proportions reflect each cell type's role in immune defense. Some are always on patrol, while others are deployed only when needed. Inside the bone marrow, large reserves of neutrophils are held in readiness, poised to respond quickly when infection strikes. Once a signal is received, neutrophils are rapidly released into the bloodstream. They travel to the site of infection, where they pass through the blood vessel wall and enter the affected tissue. There, they perform their primary function, engulfing and killing invading bacteria. Neutrophils are efficient and aggressive, but short-lived. After completing their task, they die at the infection site. Macrophages then move in to clear the battlefield, engulfing and digesting the dead neutrophils and remaining debris. This process represents a powerful early wave of the innate immune response, a fast, sacrificial strike designed to contain the infection before it spreads. Macrophages are essential players in both immune defense and communication. On the left, we see a macrophage using surface receptors to detect and bind bacteria. Once bound, the bacterium is engulfed into a compartment called a phagosome, which then fuses with a lysosome to form a phagolysosome, where the microbe is degraded. But macrophages don't just destroy, they signal. On the right, bacterial components bind to specialized signaling receptors on the macrophage's surface. This triggers a cascade inside the cell that activates gene transcription in the nucleus, leading to the production and release of inflammatory cytokines. These cytokines alert and recruit other immune cells to the infection site, amplifying the response and ensuring a coordinated defense. Immunoglobulins and T-cell receptors are the diverse lymphocyte receptors of adaptive immunity. Adaptive immunity relies on two key types of receptors: immunoglobulins, found on B cells, and T-cell receptors. B cells express surface immunoglobulins, or B cell receptors, which recognize antigens directly. When activated, they can be released as antibodies, circulating freely in the body. Both the surface-bound and soluble forms share the same structure, with variable regions at the top that bind specific antigens, and constant regions that remain unchanged across classes. In contrast, T-cell receptors also have variable and constant regions, but they stay membrane-bound and only recognize antigens presented by other cells. This combination of structural stability and antigen-binding diversity gives adaptive immunity its precision, flexibility, and memory. On encountering their specific antigen, B cells and T cells differentiate into effector cells. Antibodies bind to pathogens and cause their inactivation or destruction. Antibodies are powerful tools of the adaptive immune system, not only marking invaders, but actively helping to eliminate them. One method is neutralization. Antibodies bind directly to bacterial toxins, preventing them from interacting with and damaging host cells. The toxin-antibody complexes are then engulfed and destroyed by phagocytes. Another key process is opsonization, where antibodies coat bacteria in the extracellular space. This coating makes the bacteria more recognizable to phagocytes, which ingest and destroy them. In a third scenario, antibodies work in tandem with the complement system. Complement proteins bind to antibody-coated bacteria, forming a complex that's even more attractive to phagocytes. Specialized receptors on the phagocyte surface recognize the complement-tagged target, ensuring its rapid ingestion and breakdown. Through neutralization, opsonization, and complement activation, antibodies don't just recognize threats, they help remove them with precision and force. Most lymphocytes are present in specialized lymphoid tissues. Lymphocytes, B cells and T cells, aren't scattered randomly throughout the body. Instead, they're concentrated in specialized lymphoid tissues designed to detect and respond to infection. Primary lymphoid organs include the bone marrow, where B cells develop, and the thymus, where T cells mature. Once developed, lymphocytes migrate to secondary lymphoid tissues, where immune responses are initiated. These include lymph nodes, which are stationed throughout the body and filter lymph for signs of infection. Other critical sites are the spleen, which monitors the blood for pathogens, and mucosa-associated lymphoid tissues, like the tonsils, adenoids, Peyer's patches, and appendix, which guard the entry points of the respiratory and gastrointestinal tracts. Lymphatics and vessels like the thoracic duct link these sites, allowing lymphocytes to circulate, communicate, and coordinate a rapid defense. This network forms the surveillance system of adaptive immunity, constantly on the lookout for signs of danger. Lymphocytes are constantly on the move, circulating through the body in search of signs of infection. They travel through the bloodstream, exiting through small vessels into lymph nodes. Inside these nodes, they scan for antigens presented by specialized immune cells. If no threat is detected, they exit the node via efferent lymphatic vessels, which return them to the blood through larger ducts, completing the loop. This constant recirculation allows lymphocytes to efficiently patrol the body, sampling lymph from tissues, maintaining surveillance, and quickly mounting an immune response if a pathogen is detected. It's a dynamic, continuous journey, ensuring protection is never far away. Adaptive immunity is initiated in secondary lymphoid tissues. Naive lymphocytes travel through the bloodstream, arriving at lymph nodes via arterial blood. Meanwhile, pathogens from infected tissues are carried into lymph nodes through lymphatic vessels. This is where the immune system springs into action. Lymphocytes encounter antigens, become activated, and begin mounting an adaptive immune response. Once the response is underway, lymphocytes and lymph fluid exit the node, re-enter the bloodstream, and continue circulating, ready to defend the body wherever needed. Inside the lymph node, antigens from lymph enter through afferent lymphatic vessels and flow into the marginal and medullary sinuses. Lymphocytes are organized into specific regions. T cells gather in the paracortex, or T cell area, while B cells concentrate in lymphoid follicles. When activated, B cells form germinal centers, where they proliferate and produce antibodies. The filtered lymph exits through efferent lymphatic vessels, carrying immune cells ready to defend. Lymph enters the lymph node through afferent lymphatic vessels and flows through the marginal and medullary sinuses. Within the node, T cells concentrate in the paracortex, while B cells are located in lymphoid follicles. When B cells are activated, they form germinal centers for antibody production. The cleansed lymph exits through the efferent lymphatic vessels, along with activated immune cells ready to defend the body. This stained lymph node section highlights key immune regions. The lymphoid follicles contain mostly B cells, with germinal centers appearing lighter due to active proliferation. Surrounding areas rich in T cells are found in the paracortex. In the center, the medullary sinuses serve as channels for lymph flow and the exit of immune cells through efferent vessels. When bacteria enter tissues, macrophages and dendritic cells respond. Dendritic cells carry bacterial components to nearby lymph nodes via afferent lymphatic vessels. Inside the node, they present antigens to naive T cells, activating them. These T cells then help B cells become plasma cells, which secrete antibodies that exit via the efferent lymphatics to fight infection. The spleen provides adaptive immunity to blood infections. The spleen filters blood and mounts immune responses against blood-borne pathogens. The red pulp removes old red blood cells, while the white pulp contains immune cells. Within the white pulp, the central arteriole is surrounded by T cells in the periarteriolar lymphoid sheath. B cells form follicles with germinal centers, where they become activated and produce antibodies. The spleen is a critical organ for adaptive immunity to blood-borne pathogens. It contains two main regions: the white pulp, rich in lymphocytes, which initiates immune responses, and the red pulp, which filters and removes old or damaged red blood cells from circulation. The white pulp of the spleen is organized around a central arteriole and contains distinct immune zones. T cells cluster in the periarteriolar lymphoid sheath, while B cells form the B cell corona around germinal centers. Surrounding this are the marginal and parafollicular zones, which help filter antigens and initiate adaptive immune responses. These images show how the spleen responds to pathogens in the bloodstream. The left image displays antibody-producing cells clustered in distinct foci after exposure to a blood-borne antigen. The right image shows a spleen without those active germinal centers, highlighting the importance of adaptive immunity in fighting systemic infections. Most secondary lymphoid tissue is associated with the gut. This image shows how the immune system protects the gut. Specialized M cells in the epithelium transport antigens from the gut lumen to immune cells beneath. Dendritic cells, T cells, and B cells are organized into follicles with germinal centers, where immune responses are initiated. These lymphoid structures drain into efferent lymphatics, linking the gut to the broader immune network.